Guys, I'm glad the weather cooperated. This is They quieted down next door. Well,
all right, it is May 11th, 2026. It's a Monday, and you're watching the Biotech Voyager. It's the show that covers what's next in biotech, and today I was able to link up with John Boyce over at 28 Capital, and he provided this incredible venue where you can see the Boston landscape in behind. Really excited to be covering what's going to be happening at ASGCT this week. Thank you everyone for coming. We're going to be covering a variety of different things here today. We're going to start by overviewing some of the different sessions, some of the key posters that you should be watching. But then additionally, I've got John Boyce here off camera. He's smiling at me over at 28 Capital. He'll come in and provide his perspective on the gene therapy landscape. And we also have Chimin Kwan from NanoMosaic who will be coming in as well to join me to talk about their really cool technology and also provide some of his thoughts on where our gene therapy space is coming. So once again, thank you everyone for coming. I'll try my best to keep up to date with the comments here. But thank you everyone for coming. Excited for this. If you have any questions that you would like to ask folks, feel free to put them in the chat and we will get to them as quickly as we can. All right, so right off the bat, where we're going to start here, we're going to start by just kind of overviewing some of the key ASGCT company announcements. So, right, ASGCT, the American Society for Gene and Cell Therapy, focuses on, it's one of the biggest shows of the year, no doubt about it. And in particular, what we want to focus on here is that companies will come to this event and they will release some of their really big announcements just prior to the show starting, so they generate a bunch of buzz. So we're going to go through some of those different ones here and just kind of talk about them. So we'll plan on doing that, covering some of these different ASGCT company announcements. If you have any that you have seen that you want to talk about, feel free to put a comment down below and we'll try to get to that. And we'll be off to the races. Okay, so we're going to start here just kind of with a bit of an overview with regards to what we've seen so far. There's a couple of different strategic themes that we want to focus on. So first of all, in vivo CAR-T is one of the dominant stories for this year. It's been generating all sorts of buzz. Lots of different investment has gone into the space. So that's one of them. Another one is also the fact that AAV gene therapy, we're seeing platforms, yes, but we're also seeing a lot of really exciting clinical readouts as well. So that'll be another key theme that we'll go through. Additionally, we're seeing capsid delivery and capsid and delivery engineering consolidating around AI, right? AI is really going to be able to come into this component of the application, designing the capsids themselves for gene therapy applications. So we'll be able to talk about that. Manufacturing, obviously, gene therapies and cell therapies. There's a plane. See the plane? Gene therapies and cell therapies, very expensive to make. And so manufacturing these is one of the key components that's going to ensure whether or not we're going to be able to get these to the level that they need to get to. And so we'll be looking at some of the manufacturing infrastructure as well. And then number five is RNA medicines and the fact that they are diversifying beyond the liver. RNA obviously kind of got its comeuppance around COVID, for better or for worse, depending who you're talking to. But in particular, we are seeing some really cool applications coming out there with regards to therapeutic applications, all sorts of different stuffs. Okay, so we're going to start here just with some of the key signals to focus on. One of the ones that I talked about last week was Lattice Bio. So they raised $97 million, a Series A led by SVC, and they're expecting a clinical readout later this year, and they're also going to be going after Huntington's. So a lot of really exciting applications there for sure. Precision Biomedicines. They got approvals in France and Romania for an HBV trial. So that's going to be really exciting to watch this week. Another one here from Oxford Biomedica, one of the premier leaders for CDMO applications of viral vectors. They have just launched a fast-track CDMO service where you can get your AAV production, your entire batch, in 7 months. That's pretty big compared to kind of the status quo. And then Dino Therapeutics, one of my favorite companies to watch inside of the space, just announced a second AAV capsid license with $15 million plus in milestones for skeletal muscle. So again, AAV has really has had a real challenge in some of these different delivery areas. In particular, Dino Therapeutics has been able to come in and design some really powerful capsids that are able to detarget the liver in a way that a lot of different natural capsids and engineered capsids can't, and really specifically focus on that particular skeletal muscle that you want to go after. And the fact that Astellas is licensing another one is a big deal that hopefully I can meet up with some folks from Dino Therapeutics this week. I know a couple folks over there, and we can learn a little bit more about it. Okay, the next thing here. Aurion Biotech, AURION. In particular, they have their first patient dosed in a phase 3 for corneal edema. All right, phase 3. There's been lots of doom and gloom across the AAV gene therapy space, lots of different areas where people can come in and sit and be naysayers. But in all reality, we have some really promising late stage gene therapies that are coming around. And this is one of them. They've been able to dose their first patient for a pretty important disease. So we'll definitely want to keep an eye on that going into the week ahead. All right, there's just a couple comments here. Let's see. Cercio, yes, we will. we're apparently a lot of people want to talk about Cercio. So if there's something in the chat that we should specifically talk about, please let me know and we can get to it. I don't think I have Cercio in this particular list. So by all means, let me know what we should be talking about and what we should know about for this one going into ASGCT. By all means, let me know. Okay, we're going to go from that one. Well, before we go any further, folks, just in terms of what we're planning on doing for the rest of the hour that we have together, we're going to continue to go through some of these key company announcements. Secondly, we're going to be focusing on some of the different posters and some of the different sessions that you should go and see. I made some LinkedIn posts about that in the last couple days, so we'll kind of revisit some of those. But secondly, we do have a VC here. He has provided this beautiful Boston backdrop for this live stream. So we're going to be bringing him in to talk about the gene therapy space and kind of where he sees things going. There's another plane. Look at that. Boston Airport is there. It's over there. Yeah. So everyone's here to listen about Cercio. All right. So please let me know your thoughts about this for sure. Okay. So moving on from there, we want to quickly take a look here at AI-RNA, okay? So this is a company called AI-RNA. I wonder if they're using AI. I'll bet you they are. And in particular, they have a first patient dosed in a phase 1 of AIR-001 for AATD. AATD, what does that stand for? But it has an FDA orphan drug designation. So it's exciting to see some of these different RNA therapeutics coming along and providing a substantive addition to the gene therapies market. So that'll be exciting to watch. Historically, right, when it comes to gene therapies at ASGCTs that I've been to in the past, you've mostly focused on AAVs, a little bit of lenti, some LNPs that are delivering RNA. But it is interesting to see how that part of the field has really risen in a meaningful way. Yeah. Okay, so AI-RNA, definitely going to be looking around for that one this week. Another company called Sensorion. All right, so audio gene phase 1-2, 6-month interim, early efficacy and dose response in OTOF deafness. Okay, this one's super interesting. OTF is something that I think we looked at a couple weeks ago, where essentially you have the hair cells and they are kind of vibrating with regards to the sound, but then they aren't able to actually send those audio signals through to the brain, and OTOF comes in. I think this is a similar one that Regeneron just did. All right, yeah, okay. I've got confirmation from Chimin. Same genetic disorder there. So again, another follow-on. Hopefully, some really encouraging data there. Okay, we've got a couple more here of the key ASGCT company announcements to focus on, and the next one here is going to be Azitra. Okay. They just recently raised $10.5 million placement with up to another $31.4 million in warrants. So again, there is some stuff coming into the space here. Okay, Cercio platform. Okay. Thank you. Kenobi-ish10 finally put in a little bit of additional component here. Cercio develops next-generation circular RNA technology, CircVec, for gene and cell therapy, showing 40 to 50-fold stronger expression compared to standard mRNA, and is partnering with global pharma. And they will be presenting an abstract. Perfect. Okay, this is sweet. I think that might have, I'm not sure if that made it my list of different oral abstracts or not. So if it did and I'm forgetting, that's on me. But yeah, 40 to 50 times stronger expression than standard mRNA. That's awesome. Okay. And they're going to be presenting an oral abstract. So if someone can put the details for that in the chat, ideally everyone else can do it too. Yeah, Lene Mary Carlson is saying you need to do a deep dive into Cercio and their new CircVec platform and report straight to us. Thank you. Well, I can try to do that. Maybe what we can plan on doing is if any of you guys have a contact over there, maybe we can do a video together at ASGCT, or have a dedicated episode for them here on the Biotech Voyager once the craziness of ASGCT is over, or we can do both. We'll see. Excellent. Okay, so we're almost through the ASGCT company announcements. Next up, we'll have John Boyce from 28 Capital, but we'll just get through a couple more of these announcements, and then John will come on. So stay around for John over at 28 Capital. All right, Sangamo Therapeutics is another one that it'll be interesting to follow as well. They have a rolling BLA underway for ST920 in Fabry disease. So definitely will be interesting to see what data they've got around that. Another thing that I want to point out that just happened this morning, Pompe disease just got a phase 1 trial from AskBio. So AskBio, started by Jude Samolsky, I think back in the early 2000s, Jude Samolsky being one of the huge pioneers inside of the space. He very recently this morning, AskBio announced data that they have they've dosed their first patient for an AAV gene therapy that is going after Pompe disease. Pompe disease, go look it up, but it's a pretty rough disease. I know there's some good work as well going on down at the University of Florida, I believe, on Pompe. Definitely a key disease that deserves a lot of love. So it's great to see some different applications going after this. Yeah. Okay. So guys, if there are any other companies here that we should be talking about, by all means, put them in the comments. Cercio has gotten a lot of love, so we're gonna have to look these. We're gonna have to do a deep dive later on for sure. All right, so we'll stop there for now with regards to some of these different company announcements. If there are any other ones, please let me know, and we can highlight them here in the chat. And we will we'll get back to that. Okay, so we're gonna shift gears here just a little bit. We've gone through some of the key ASGCT company announcements, and we're just gonna move on here to John Boyce over at 28 Capital. Give me 1 second here. I'm just gonna put it up on screen here so everyone else can see. All right, so John Boyce from 28 Capital is in the house. In fact, this is his deck. So John Boyce, welcome to the show.
Thanks, man. Thanks for inviting me to your office outside. Yeah. Very much appreciated.
All right, you'll need this.
Okay.
You can just hang on to it, or you can clip it on.
I'll
whatever
hold on
works.
this little bugger here.
All right, can everyone? All right, we'll just change this a little bit so that everyone can see John, and we can still see the beautiful background here. There we go. Okay. Perfect. John, how you doing?
I'm doing well, Ben. Thanks for coming up here. Appreciate it.
It's a beautiful day, a beautiful view. I don't know how you get any work done.
This is a little challenging at times, no doubt about that.
Excellent. Okay, folks. So again, this is John Boyce, principal over at 28
Managing Director and co-founder.
Perfect. Managing Director and co-founder of 28 Capital. If you have any questions for him, feel free to put them in the chat. There are obviously some things he can't say, but if you have any questions for a VC inside of the gene therapy space, please do let us know. All right, so John, obviously you guys have focused quite a bit on AAV, gene therapies in the last couple of years. I'd be curious to kind of get your perspective on where the space is going from here.
Yeah, thanks, Ben. I often get asked the question, is there a bottleneck in the AAV gene therapy space, and is the field broken, and can it actually scale? This is a common recurring question that comes to me quite often. And my answer to that is that I don't think the gene therapy field is broken. In fact, I know it's not. It's just under repair.
Right.
You know, we have world-class biology being supported by a tech stack that is straight out of the 1990s. So unfortunately, we need to get that up to par to enable some of these great breakthroughs that are coming forward. You know, and the numbers say it all, actually, from an investment standpoint. In 2021, there was $8 billion that went into gene therapy venture across 122 rounds of capital. 2021, it went down to 1.2 billion across 39 rounds. But we see that coming back. And I used to have an expression that pharma and investors value the golden egg more than they do the golden goose, meaning that, you know, you would talk to an investor, you could talk about your great platform that could actually help the gene therapy stack back in the day, and they would just say, yeah, you know, bugger off, come back and talk to me when you have a candidate that is ready for clinical trials. Well, you still need that. That's still important. But this day and age, whoever actually has that tech stack and can fix that is going to win. First and foremost, the manufacturing aspect. You were nailing it too. Yeah. When we look at an opportunity, we no longer ask the question, will your drug work? It's, will your drug work, and can you actually make it and make it cost effectively? So that is something that comes up. And the thing we see a lot is full versus empty capsids. Can you make that efficiently? Can you get the cost down? So there's technologies out there that are controlling the producer cell line, something we've been spending a lot of time doing. And we have one technology in-house, and the goal is, and we're showing promise of this, to get the cost down to, you know, for example, if Zolgensma now for $300,000 a dose, down to about a little under $1,000 a dose. And that, when talking about the whole population that you're treating on that, it takes it from $1 billion to treat the entire population of Zolgensma down to about somewhere around $3.1 million. So these are the technologies that are coming up. So can you actually make it efficiently? Can you make it cost effectively? And then safety. You also mentioned retargeting. Retargeting is something that's very hot and very much on our radar at this point. We've seen technologies doing that. Dino's doing a great job at that. There's others out there that are doing it. We have a company, Brick Bio, that's targeting very well. We published in Molecular Oncology in 2025. It came, you know, we targeted AAV2 and delivered 85x more to the tumor, as opposed to just the capsid itself, and 45x less to the liver. So I mean, there's technologies like that coming out. There's a lot of good things that are coming out along that path. So not nailing that down, but I always say that you can't fix what you can't see. Yeah. And so how do you actually, the analytics has been something we focused on, and we've been spending a lot of time on. Hence is why we have our next guest coming up here, NanoMosaic. It's getting a little windy up here. You okay, man?
Yeah, all good.
All right. Microphone's good? Or?
Oh, it's
okay.
Good. Yeah. Okay. Just the other computer turned off for a second here.
Okay, no worries. So, you know, can you actually, you can't see what you can't fix. And that's something that we've been saying for a while. And I mean, Peter Marks was talking about this back in the day, that, you know, do we really know the empty full ratios of the capsids themselves? We have a technology which Chimin will come and talk about, which we have we put a lot of faith in, and it's measuring both transgene and capsid on the same platform at the same time. And what we found is shocking. We've seen some companies, large pharma from CDMOs at the manufacturing stage, say that they're 93% full. When they'll do ddPCR, maybe ELISA, maybe mass spectrometry, they're counting their partials, and the normalization between the different technologies with their own inherent biases doesn't work out. So what's the answer? We've seen that they're adamant that we're 89% full, 93% full. They're actually 3% full.
What? Well, okay.
It's shocking.
Stop for just a second.
Chimin will reveal more on this one, my friend. But
so AAV, you've got someone who thinks they're 90% full.
Yes.
90% full. They run it via, qPCR or AUC or whatever else.
You got it.
They get all those readings back. We're at 90% full, so that means we've got 90% therapeutic product all the way down, and it's 3%.
3% at the manufacturing stage. And Chimin will go into the pipeline a little bit more.
Oh dear.
So we presented all this data to the FDA back in July, and we're hoping for FDA approval at some point. We were figuring it could be some time. We knew this was on their radar, something they were proactive at fixing. We got FDA approval. We got the AMT grant within 6 months. So it actually, tens of thousands, hundreds of thousands of data points we provided them on this one, showing that how we compared, how we did orthogonal methodologies to measure it, but it came across. So this to me is the ground truth. What are you actually measuring?
Yeah.
And what is the true measure of that? And then how do you go about fixing that? So that's the first thing that we focus on. But the rest of the tech stack needs to be fixed as well. But we see promising technologies. I think this is the year to invest in gene therapy. This is the year it's coming back. And we're really excited about it. And we're actively looking at opportunities within this, both from a tech stack perspective, as well as from a candidate perspective as well.
Perfect. Okay, sorry about that.
No worries.
The second computer just turned off for a second.
Blew away. No,
Chris has yikes, 3% full. So yeah, I first met John when I was working at NanoMosaic a couple of years ago, and yeah, it's fascinating, right? When you're trying to build trillions of gene therapy vectors, it's who knows? It's kind of hard. And so it's always a good idea to have good analytics to figure out whether or not you're going down the right direction or not
100%.
when you're making trillions of something over and over again. And you got to make them at a high quality.
100%.
Yeah, wild. Like John, I would again, folks, if you have any questions for VC inside of biotech, feel free to put them in the chat. But like John, I'd be curious just to kind of get a little bit of a background on like your story. Like, how did you get into the space? I don't know. You haven't really prepared for this, but I'd love to learn a little bit about kind of where you came from and how you've gotten to where you are.
Okay, it's great. I will do my best to fill in again. Not prepared. Ben just showed up at my house today and just came up my roof deck. I had no recourse. But
I was invited.
No, I'm kidding. Of course. I'm a serial entrepreneur. I've done a number of companies, exited from there. Always loved next generations of technologies and trying to figure out what's next and what's going to make an impact in human health and how we can actually better that population. And I love to give back. I love to give back. And actually, this was a way of actually having a bigger effect in the fields. Taking young entrepreneurs and companies that are coming up, we invest in the earliest stages. In other words, we were first doing technologies that were stuck in academia, taking them out of academia, and that's where we got NanoMosaic from. That's where we got some of our other companies from, and working with the founders, teaching them how to get the intellectual property out, and then build a company around that, and then funding it and getting it to the next stage. I'm also a fellow at Harvard. I teach entrepreneurship and innovation. The class has grown from 8 to about Harvard. 420 now. Yeah, so they let me in there every now and then. So nice. It's like. It's been. Can I come sometime? You absolutely can come, Ben. 100%. You can give a talk.
I haven't been to Harvard yet.
But yeah. So I'm just driven and excited about the next generation of technologies that are coming up. What's going to have an impact in the field, and across human health in general. So this is really something that I've been focused on and love working at that early stage, later stage too, because we have to get returns, of course, being a venture capital company. So it's a mix between the two, but a lot of VCs were really worried about the early stage. We focused on that and we went hard on that in the beginning, and we still stand our ground on that. So would love to talk with all of you and follow up from there.
Fantastic. Sweet. All right. Everyone, say if you can, say thanks to John in the chat or something. I'm new to this live stream thing. I don't know how it works. W's in the chat. W's in the chat for John. I don't know what I'm doing. All right. Awesome. Thanks so much, John. Yes, that would be great. Okay. Let's have a chat here with Chimin.
Thank you, Ben. Thank you, guys.
So thank you. All right, Chimin, you're up next, and I'll probably get you to just open up your computer to LinkedIn as well. But you'll need this. Okay. You can either put it on your lapel, or you can just hang on to it. Whatever works.
Sounds good.
Excellent. Okay, you'll probably need to bring it up a little bit higher.
Okay.
Sounds just good. So we make sure that everyone can hear you. Yeah. Excellent. Okay.
I'm looking at myself. It's very weird. Black eye.
That's all right.
Stayed up really late last night.
All right. So we're gonna move on here to Chimin, but just a second. Wait a moment. There's a question here from Chris for you, John. So maybe you can just answer off. You mentioned 2026 is the year to invest in gene therapy. what's changed as the funding climate has been tough in the last couple of years? Is this a reflection of improving macroeconomic factors, or is this more there are now undervalued assets to be had? Are you able to talk about that?
Excellent. No, excellent question.
Okay, can we give that back? John, you come back in for a second. Okay. Sorry, Chimin. Great question, Chris. Thank you. All right, we'll move this back to John Boyce answering your questions in the chat. Thank you, Chris. Okay,
Chris, thank you for that question. Excellent question. I think in years past, as I keep talking about this tech stack, we've seen things that are actually really bringing the cost down to manufacturing, really getting retargeting working in a way that wasn't working before. So now that this tech stack coming about, I really see this as a breakthrough moment. So actually finding assets that utilize this tech stack, that utilize the analytics, it's going to make a path forward on this. So, I mean, there is, some people say it's still risky at the time, but this is where you actually, from an investment add-on, this is where you make the biggest gains, actually. So taking the chance, working with people that are driven, working with smart entrepreneurs that know how to drive a product forward with the tech stack that's enabling it, I think that actually goes a long way. Yes, the macroeconomic is still a little bit challenging, no doubt about that. There's light at the end of the tunnel. We see that starting to happen, but I think for the right therapeutics, the right everything good?
Yep. Sorry.
Okay, no worries. The right tech stack behind that, I think we're going to get there. And again, there's some things I can't talk about in terms of what's happening in manufacturing, other than what I spoke on before and retargeting. But again, really excited, really jazzed what I'm seeing. And I think that there's a number of things that people are taking this tech stack build more seriously now. So.
Fantastic. All right, John back with another question.
Thank you, Chris. Appreciate it.
Thank you, guys. All right, now we're going to bring in Chimin for real this time. Chimin, you've been a gem. All right, there you go. Perfect. Okay, we're just gonna. We're just using a bit of an ad hoc system here to make sure that everything stays stable. Okay, there we go. All right, Chimin, perfect. You are the co-founder of NanoMosaic. We first met a couple years ago, was able to help you launch the Tessie device, so we can definitely get into that for a little bit. But first, I would love to, again, just kind of get a little bit of background on you. How did you get you started inside of gene therapy? How did you develop the technology that underpins Tessie? Tell me your life story.
Yeah. Thank you. Yeah, I promise you, I won't go from when I was born, from there, but I was really trained as a physicist. So I did my PhD at Harvard. Initially, I really hate biology. To be honest, I have to confess that a little bit.
Well, can I make a confession to you?
Please.
I loved biology and hated physics.
Okay, all right, we're good partner there. Yeah.
We're like hand in glove. There we go.
but I soon realized there's a lot of opportunities in modern biology, especially coming from a physics background. I'm trained in more quantitative science, so a lot of tools that we use in, for example, nanotechnology, quantum information, optical communications, that can be used to develop novel platforms to solve biological problems. So I, after PhD, kind of got really lucky, get a PI position at Harvard and geared my research towards developing platforms to solve very pressing healthcare problems. So one of the first things we're trying to do is scalation.
Yep.
For example, if you're talking about diagnostics, right? So it's so expensive, and we're looking at so less markers. Yep. So the company NanoMosaic was really founded from a concept that we can leverage a CMOS platform, nanophotonics, optics, to really decrease the cost of omics. And then the company developed, we were able to really find this opportunity in cell gene therapy where during the manufacture process, there are so many attributes to measure, and usually those are multi-omic attributes. For example, you need to detect capsids, you need to detect proteins, you need to detect transgenes, DNA, RNA, potency assays, right? So all of those, we have a really unique way to quantify using
Fantastic. Sweet. Cool. Yeah. So I guess just kind of take me back through how this technology works, right? So you have the ability to take a sample of AAV, you can analyze it, and from that sample of AAV, you can give me the full, partial, empty ratio for AAV gene therapy.
Exactly. Yeah, I think you summarized really nicely. But the opportunity we see here is the gap between analytics to the potency assay, right? So we measure all these attributes, right, capsid titer, empty full ratio, but still after optimization, we don't see a lot of infectivity, efficacy at the downstream, right? So where's the gap? What we found is we're using a lot of proxy when we do the measurement. Empty-full ratio, for example, right? So we're using ddPCR, qPCR. We're only looking at a small fragment in the transgene, right? Those fragments, especially in the context of our AAV, it's usually not full length, right? So what we're doing here is accurately, specifically measure the full length using our technology and build that into a multi-omic platform, so that just not only you can measure the transgenes, the full length transgenes, but also you can put other attributes on the same platform, right?
Okay.
The challenge in the AAV QA/QC, I think, are threefold, right? One is the genome integrity, empty-full partial ratio, right? So you have to have the specificity, the resolution. The second is measure those attributes in the upstream, include sample, right? Those have been challenging. We have a lot more technology you can use in the downstream, but not a lot in the upstream. Right. And the third one is really throughput automation, something you can harmonize the whole manufacture process, right? The same analytics you can do in the upstream optimization, but also carry the same thing across the manufacturer, so you get a unified analytics out of it.
Yeah, and again, maybe just to speak to the point that John discussed earlier, right? A lot of different people who are designing an AAV gene therapy, they come in and they've designed their process and they go and do their titering of their AAV, and they're like, hey, we've got 90% full. Good to go. So that means 90% of the vectors that we've made are going to have the therapeutic gene inside of them that we can then stick inside of patients, and we can calculate based off of that. And it's actually 3%.
Yeah, I would say there are a lot of room in the AAV manufacturer that we can improve, right? For example, we evaluated probably 20, 25 different programs, upstream to downstream. Yeah. I think the most intriguing thing what we're seeing is the gap between the true full length versus what's characterized using compendial technology. For example, like the extreme case is in the upstream. I think ddPCR, ELISA, right? So they gave roughly 90%. Yeah. But when we really measure the true full length, it's only 3%. Having said that, this is very upstream, right? So haven't gone through purification optimization, right? But still, that tells us there's a lot of opportunity. Think, what if you can, in the upstream, after the clone selection, you can get it from 3% to 30%, 90%, right? Then potentially you can save 10x of bioreactor volume. You can drastically decrease the cost.
For sure.
Right? And also you can also change your dosage, so it will be more toxicity tolerant, right? I think last year at ASGCT, there was a lot of discussions on the toxicity hurdle, bottleneck, right? And the solution for that, I think we're making a lot of progress within last year. Something I think to look out in this year, I think there's a lot of discussions on building into good analytics into the manufacture process, right? So you can fix it in the upstream, and also receptor targeted capsid, right? Evolving from custom capsid, so that if you do systematic injection, right, it's more targeted, so you can deal with much less dose.
Gotcha.
So all these combined together, I think we're going towards higher quality AAV, potentially lower down the manufacture cost, be more pure, right, in terms of the final product that we inject into the patient.
Yeah, awesome, love it. Yeah, I was a big believer in the NanoMosaic system a couple years ago, and I still am. So
thank you.
Yeah, if you're making an AAV gene therapy, you probably should have one of his devices to make sure you're not getting fooled by your fulls. Yeah.
Thank you. Thank you.
Excellent. Okay.
Yeah, that's a really good way to summarize it. Don't be fooled by your fools.
That's right.
And maybe I can say a few more words. We also have a few presentations in the conference.
Please.
I would love to meet with you guys. So we have one presentation which is on Thursday.
Excellent.
We're gonna talk about our recent AMT approval from the FDA, and really touch upon what that means for sponsors, right? So what's the benefit of using a FDA AMT designated platform?
Yeah.
We also have a couple posters. We're also launching a potency assay together on the same platform, right? So not only you can do the analytics, but also you can run a potency assay on our platform.
Interesting.
We also announcing a collaboration with TCAN in terms of using their VIA system to automate our workflow, right? Gearing towards more GMP fully automated system. We also have 3 customers gonna talk about data running our platform. So stay tuned.
Very good. Okay, there is a question here from David Hayes.
Okay.
Let's see if we can put it up here on screen. Is there a reference that discusses the 90% claim when only 3% is active product? Is that something that people can find somewhere? Will there be some sort of release, or are we just, or would we have to keep that confidential for now?
So come and talk to us. We can show you some data, but unfortunately, we have to keep it confidential. You know, like we cannot disclose what programs they are. But right. I would also say this. We also evaluated currently commercial drugs like on the market, and luckily those quality is good.
Excellent.
I think the key thing here is, you know, we're spending so much money, so much time in the downstream, right? Throughout the purification, and then because the initial starting point of the drug is not high purity, right? So we have to 10x the bioreactor volume, right? So that just a lot of cost, a lot of time, right? So we're trying to fix this from the origin, right? The first principle concept, right? So we want to provide you guys with a technology that we can look at the quality attributes at the very beginning at the bioreactor level.
Perfect. Excellent. All right. Thank you so much, Chimin. All right. I will get you to turn the computer back on for a second if you don't mind.
All right.
And then from here, folks, very kindly, Chimin was able to come in and answer some questions and talk about AAV analytics. So just with the time that we have left here, let me change the banner so it makes sense. We're going to go through some of the ASGCT posters to watch. There are literally thousands of posters at the event, and you can't get to all of them. Some people try, and they might succeed, but they won't be doing anything else the rest of the day. So what we're going to focus on now, let's just change this a little bit. We're going to focus on the on some of the posters to watch. So Chimin, if you're okay with it, I'm going to open up LinkedIn and go from there, if that's all right. Sweet. All right. So yes, folks, we're going to be covering some of the key ASGCT posters to watch. And we're also going to be focusing on, just a second here. Might have to pull it up on a different screen. Just a second here. All right, we're just going to pull it up on here. Some of the key posters that you should watch. I did a list about this earlier this week. I think I ran a competition last week where I basically asked a bunch of people, hey, I'm going to be making a list of 10 different posters. 5 of them I'm going to pick myself. 5 of them will come from people who comment down below on this post and pitch their poster. A bunch of you did. It was awesome to see. And so what we're going to focus on now is actually taking some of those posters and we're going to just highlight the ones that I found to be really valuable and that you should keep an eye on at the show. And I will try, and if you're not going to be at the show, I can try my best to highlight some of them because I think they are really cool. So one moment here, and let me pull up that list and we can get into it. Okay, so the first one here that we are going to focus on, which is actually the winner, is, or actually, let's keep the winning one to the end. And we'll kind of go through some of the different candidates from the people who actually submitted a poster for it. So the first one is from SK Farm Techo. A good friend of mine, Brian Tomkowitz, he pitched this one, doing some really good work inside of the gene therapy production space. He kind of looks like John Cena. So if you see him and you yell John Cena, he might be happy, he might not, I'm not sure. But in particular, he has a poster 2176, and they have been able to develop a scalable lentivirus purification platform. So loads of different applications use lentivirus inside of gene therapy and cell therapy, but being able to get a lot of it is really hard, not only in producing it, but then once you have it and you've got it in this slurry, in your slop, it's really hard to get those, to actually get them out of purification. And so he has teased, he hasn't given me a whole lot of details yet, how they've been able to develop a platform that basically blows the socks off anything they've seen so far for taking your lentivirus and being able to take it and purify it to at large scale with an impurity clearance that's second to none, according to Brian. So I look forward to learning more about that later this week. Okay, number 2 on the list of the top 5 for contest submissions. Well, Yeah, this would be number 2, but it's not the winner, but still very, very good. Gamma delta T cells for glioblastoma from Innate Bio. Gamma delta T cells, they don't get a whole lot of, they don't get the same sort of love. They're a bit less known inside of the space. They're a bit of a rarer cell type. But gamma delta T cells have a lot of promise because compared to normal CAR T cells, when you think of making like a cancer treatment or something, they don't have the same problem with cytokine storms. They've got a different series of weapons that they can use that are a bit more refined, that don't have quite the same challenge when it comes to overreacting with the immune system. So really excited to see there. So looking forward to them. So that's poster 328 from Innate Bio. All right, another one. Again, I'm inside of the AAV space. Well, at least that's where I got my start. I cover gene therapies and cell therapies and other kinds of biotech. But in particular, there was this group called GeneMedi, GeneMedi, and they have developed a purification system for AAV that instead of using something like an ultracentrifuge that costs hundreds of thousands, well, maybe not hundreds of thousands, but tens upon thousands of dollars to purify your AAV, they actually have an affinity spin column you can stick inside of your normal centrifuge that purifies out the AAV in under an hour with better infection efficiency than normal stuff. That's a big deal. Again, downstream purification from my days in the lab was it sucked. I hated it. It was the worst part of my job. Not that I didn't love my job, but that was the part I enjoyed the least. And so it was difficult to get all the different tubing set up and to make sure you had good flow and you did your buffers all correctly. These folks have developed a spin version, so I'm curious to see what that looks like. So that is poster 3164, and that's from GeneMedi. So congratulations to them. One more, and then we'll get to the winner. 3373. Now this one's kind of outside of standard manufacturing or therapeutic applications. This one's kind of a bit more of a meta strategic one. And so this one comes from Cencora, Cencora, and they're dealing with payer data gaps for cell and gene therapies, where payers can't get clinical and cost data early enough to make coverage calls on incoming cell and gene therapy approvals, right? This is something that's a big problem inside the field. People will spend a whole bunch of time making a cell therapy, they get it approved, or they get a gene therapy approved, and then they go to the market, and they think that as long as you get an FDA approval, then you're good to go. But it turns out that's just half the battle. Now you've got to figure out how payers work and how to get patient reimbursements all set up, and it's a mess that can really reduce your opportunity to get to revenue generation faster. And so this group in particular is focusing on how to basically provide that long-term clinical data as early as often to health plan decision makers. Basically, do the work ahead of time of getting set up with these different people inside of the payer space, inside of the insurance companies, give them the clinical data super early so that they get excited, so that when you go to market approval, you don't have to start from scratch. You've been doing this while you've been doing your clinical trials, and they get to see that data and they're really excited about it. So those are 4 different posters that you should look at, but the winner chicken dinner from Mike Cioffi from the University of Miami is looking at enlarged AAV capsids. Okay. To all drawbacks, right, is it's pretty small. You can't fit a whole lot of stuff inside of that sucker. And so this is what's really wild about what Mike has been able to do. You've got this 4.7 kilobase cargo limit in AAV. He has done a bunch of redesigned capsids using all sorts of computer modeling and stuff like that. And he's been able to design an in silico version of AAV that is 5 times the size. It's able to, or rather, it's able to package 5 times as much. It takes the cargo pack capacity of AAV from 4.7 up to like 25 kilobases. 5 times the size. It's wild. So I got to see this one. Super fascinating. And yeah, Mike, you win. Good job. I would love to see you at the conference, and I look forward to stopping past your poster, and I'd love to make a video with you. So yeah, look forward to that one. I hope to be making a video with Mike later this week. All right, so those are the ones that people submitted to me. However, I've also been going through the 3,500 long page abstract notebook, and so I've identified some other ones that I think you should pay attention to as well. All right, so the next one here is 209, and it's from a company called Sonothera. I did a post on them last week, and it kind of took off. And basically, they have developed a non-viral way to deliver very large capacities of gene therapy. Where, again, something with viral vectors, right? One of the challenges is you might be limited by size, or you might be somewhat limited by immunogenicity for repeat dosing. These folks use these like specialized microbubbles, and then a literal, and then sonar poration, which is like ultrasound. They use a combination of these like specialized microbubbles and ultrasound to basically deliver large genetic payloads inside of cells, where you basically have these microbubbles going around in your blood, and you apply the ultrasound to one particular spot, and whenever those microbubbles come past, they kind of like vibrate a certain frequency and like pop, and they'll like go inside of the cells. And the benefit being here, you can deliver a very large gene with very little issues with regards to like immunogenicity, because there's not really a whole lot on these microbubbles that the immune system can pick up on. So it could be a really interesting addition to standard viral vectors, LNPs. This is kind of like a third option for gene therapy. So I'm really excited about them. I've been following them for a really long time. Another one is Lattice Bio. So again, I mentioned this a little bit earlier in the stream, but Lattice Bio is raising a boatload of money to go after Huntington's inside of the gene therapy space. Huntington's is a disease that afflicts a couple people in my extended family. I don't carry the gene, thankfully, but that's just something to kind of keep in mind. There's a plane going overhead. Beautiful day here in Boston with this gorgeous background. But yeah, Huntington disease, a really difficult one to touch, and so exciting to see there. So how can we? I'm just seeing some of the comments here now. Sorry, I was reading on a separate screen here. How can we see him if he looks like John Cena? All right, Bart 170, you win the comments today. That's hilarious. It's a good question, but it can be done. Okay, and then Sushma here on LinkedIn as well. May I know the poster number for AAV 5 times more capacity, or the group working on AAV large delivery? Yes, you may, Sushma, for sure. Just a second here, let me pull it back up. That is poster, wait for it, poster 1019. You can find this on LinkedIn. I'll put the link for it in the LinkedIn event page here. I can't do it right now just because I'm in the live stream. But the poster that you want there is 1019. And it's from the University of Miami. So yeah, I'm really looking forward to it. So yeah, let's move forward here with a couple other ones. Okay. Another one here is from Trix. So this is a CAR TR1 Treg therapy for progressive multiple sclerosis. MS, most of us probably know someone who has multiple sclerosis. It's a pretty rough disease. And so it's really exciting to see a cell therapy using Tregs, which are kind of like the diplomat to CAR Ts being like the grunt army, the grunt army soldier. Tregs are more of the diplomat who comes in and like calms things down using non-violent measures. And so there's all sorts of really cool things that Tregs can do that standard CAR T cells cannot. And so very excited to see some of this work from Trix with regards to Tregs and some of their clinical data they're going to have for multiple sclerosis, which is, again, a disease that probably most of us know someone who has. And it can give a lot of hope. Okay, another one. I'm going way back in my own career. I think this is really cool. I hope you guys do too. And this one is 2175. Sorry, the one before the Treg one from Trix. That the poster number for that one is 306. Sorry about that, I missed that. Now let's go on to the next one here, which is small molecule enhancers for lentiviral manufacturing, featuring my first company inside of the cell and gene therapy industry, Biotech. Shout out to them up in Ottawa, Canada. I'm Canadian, in case you didn't know. And they are releasing some information with AstraZeneca and Sartorius. So again, these folks gave me my start in gene therapy, in the industry side of things. And basically, they have developed this panel of really cool small molecules that basically turn off antiviral defenses that are inside of cells that you use for production or that you use for your actual therapy. And basically, these small molecule enhancers will go in. They basically tamp down those antiviral defenses so that your gene of interest can either get into the cells and do a good job, or even when you are taking your lentivirus and you're wanting to transform it for a CAR T therapy application, and you want it to have a much better MOI. This kind of just sensitizes the cells, gets them ready to receive the therapeutic payload you're trying to give them. Be that lentivirus inside of a cell therapy, or even if you're making the you're making the lentivirus itself inside of your production line. The Biotech can help with both of those. They didn't pay me to do this. I just love the folks over there. So shout out to them. These small molecules can boost titers of lentivirus by 2-fold, and then also let labs cut the MOI requirements by up to 3-fold. So those are some pretty big numbers to throw around. Excited to see that poster. Okay, we have one more left, but let me just check if there's any any other comments here in the chat. All right, everyone is yelling about Circio, CIRCIO. Okay, you guys have made it abundantly clear. I've got to reach out to them. Clearly, they've got lots of fans here in the chat. So please get me in contact with these people somehow. I will try to find them. But yeah, from everything that people have said in the chat, this is something that I need to go look at for sure. Awesome. Okay, we're coming up just here near to the end of the stream, but we have a couple more posters left, and then one more segment, which will be some of the key segments, some of the key presentations instead of the posters that you should keep an eye on. So let's just go through one more here. To just kind of give us a nice, diverse overview of the different components of the ASGCT event. We've talked about cell therapies. We've talked about gene therapies. We haven't specifically talked about a modality that's quite specific to cancer, which are, or drum roll please, oncolytic viruses. Now, oncolytic viruses are an interesting application that haven't gotten a lot of attention over the last couple of years, but they actually have some pretty cool applications. And one of them in particular is from a lady down at UNC Charlotte. This is poster 3239, where she's got oncolytic virus that can kill most pancreatic cancer cells. Okay, pancreatic cancer, pretty much a death sentence. If you get pancreatic cancer, you probably have your last Christmas coming. It's bad. You might not even make it to Christmas. And so being able to address this really aggressive form of cancer is a must-have. And so this lady in particular from UNC Charlotte, poster 3239, she's developed these oncolytic viruses that can kill most cancer cells. However, oncolytic viruses are really good in this area, but there's a stubborn subsection of these cancer cells that seem to be immune to these oncolytic viruses. However, she has been able to develop type 3 interferons that point to new stratification and combination strategies. So there's, just to make sure I'm understanding this correctly, basically there are these type 3 interferons that you can exploit that you could probably use in combination with an oncolytic virus to really make sure you bang out that pancreatic cancer. So really excited to see that happen, and very excited for the show that's coming. Okay, so we've been able to cover some of the, excuse me, we've been able to cover some of the key news that some of these different companies have released ahead of ASGCT. That's number one. We've been able to talk with John Boyce, the co-founder of 28 Capital, a VC in the biotech space. We've been able to talk with Chimin, Kwan, one of the co-founders of NanoMosaic, kind of providing his perspective on the space as well. We were just able to go through a bunch of different posters that everyone should go check out at ASGCT, and I will be trying to provide some coverage on. But one of them in particular, again, the one that won from the event that I did last week, is 1019. Some wild guy called Mike down at the University of Florida has figured out how to 5x the capacity of AAV. He has been able to build a better AAV and has all sorts of in silico modeling about how this works. So I'm going to try to find Mike from from the University of Miami, and hopefully we can learn a little bit more about what the heck's going on here. Okay, excellent. So let's see. Excellent. Let's go on here just to the last section, which is we're going to focus on some of the key talks right at this conference. Most people don't think of posters when they think of a conference. You think of the talks. And so we're going to go through some of these different talks that look pretty interesting going into ASGCT. So I created an article on Substack, just kind of overviewing some of those different ones. You guys can go check it out if you look for me on Substack. But in particular, one of the things that I would like to focus on is also just the fact of just all of the different artificial intelligence that's coming into the space. Biology is probably the big, great problem for AI to fix. And there are some really cool companies inside of the space that are going after this. And so there's going to be an educational symposia that's covering all of this on Tuesday, so tomorrow, from 8 to 9:30, just kind of giving a landscape overview of artificial intelligence inside of cell and gene therapy, from discovery to patient impact. And so if you're going to be available, I would highly recommend that one. Like, that's just kind of a shoe-in. There are some key sessions first in the morning from 8 to 8:40 that look really good. So definitely consider that one. Okay, let's see what else here. We're just coming up on time. We might be able to go a little bit over, we'll see. Another one here, also at the same time, but ideally a little bit later. Will be engineering high-performance CAR T cells to overcome resistance and improve potency. CAR T cells, really powerful inside of blood cancers, haven't worked as well inside of solid tumors. So this is really going to be interesting for just kind of like the resistance and potency of these CAR T therapies, and there's some pretty powerful sessions in there. Okay, people think of gene therapies, they think of cell therapies. We're just able to talk about a good session on AI. But in particular, something that people don't really think about cell and gene therapies about for too much is infectious diseases, right? Cancer is not an infectious disease. A lot of these different gene therapies, they go after genetic disorders, not an infectious disease. But there's actually a lot of really cool things that are happening inside of the cell and gene therapy space for chronic infectious diseases, like HIV, for like organ transplant, for Borrelia burgdorferi. So like some of these different infectious diseases that where gene therapies and cell therapies can really come in. So HIV, obviously that's a big one. Being able to protect someone when they get an organ transplant, not just from their body themselves, but from any external bugs while you're in an immunocompromised position. So that's pretty exciting. Okay, and then a shout out here in particular, just with regards to effective communication. This is going to be on tomorrow. I am with the ASGCT communications group. It's just kind of a group of volunteers. And so this is an education symposia where we're going to be looking at how do you communicate science well, with regards to peer review and beyond and funding and all of that fun stuff. I like communicating science. Don't know if you figured that out. And so this is from 3:30 to 5 o'clock on Tuesday. Would be great if you're going to be there. I'll be there. So if you'd like to see me, that's somewhere where you could flag me down and say hi. So again, that's 3:30 to 5 o'clock, and that's effective communication for funding, peer review, and beyond. That's in room 052AB on the exhibit level. Okay, so we're just coming up on time here, but this has been great so far. Man, there's so many of you here in Cercio who are like, yeah, Cercio just got a 14-fold increase in its stock. Okay, what's going on here? Interesting. You guys are all spamming the chat, but this is great. Love it. Okay, so that was for Tuesday. Just here quickly for Wednesday. There's a really good session from 8 o'clock to 9:45 that's talking about emerging clinical evidence in cell and gene therapy. The long-term outcomes, right? Cell and gene therapy is still a relatively new space. These are some of the really long-term outcomes and first-in-human insights. And so we're getting to that point where we've been able to accumulate enough data and we've got enough insight that we can start to pull out some really interesting trends. So I would highly recommend going to that one. That's one that just everybody should know. That's first thing in the morning on Wednesday from 8 to 8:45. Emerging clinical evidence in cell and gene therapy. Okay, a little bit later on, engineering programmable gene therapy systems. This is going to be important, right, when it comes to being able to tune and control how much gene therapy is getting expressed, or maybe at some point you want to turn it off. That'll be really interesting. How can you program these things? And that's from 10:15 to 11:45. I'm going to grab some water for just a second because I have been yelling for an hour. Okay. There's again, Dino Therapeutics, lots of really cool companies inside the space, but Dino is really cool as well. And they've got a sponsored symposia from 12 to 1 that's covering advancing AI and delivery frontiers to empower patients with genetic agency. They've done some wild stuff, man. They had a gene therapy vector that was something like 30 times more potent, where you could get the same therapeutic effect with 30 times less dose. Wild. And so that's really going to move the needle with regards to getting a lot of people more excited about gene therapy once again. Okay, to round out Wednesday from 3:30 to 5, there's a state of the field with CBER leadership. CBER leadership, right? Terrence Flott is going to be there. One of the key bodies that's overviewing this entire field. They're going to be there. They're going to be talking about this in a fireside chat from 3:30 to 5 o'clock. You should probably go to that as well. That's where I'm planning on being, if I can, to really figure out, pardon the Canadian expression, to find out where the puck is going. All right, so that is good. If you guys have any other, recommendations of events that I should be looking at, please feel free to put them in the chat. Thank you for everyone who has participated so far. This has been absolutely great. Okay, so let's move into Thursday here. Thursday, May 14th, second last day of ASGCT. What would be some of the key ones here to keep an eye on? There's going to be a bunch of analytics, potentially, maybe featuring NanoMosaic that you should go and see on Thursday. I believe that is the next generation sequencing and analytics for cell and gene therapy and manufacturing from 10:15 to 12. I'm sure Chimin, who was on earlier, and John, who was on earlier as well, would appreciate it if you came. So definitely check that one out if you're worried about making sure that you're doing well with your AAV and it's high quality and you're actually measuring the right stuff and you don't get fooled by your fulls. Okay, let's go on here to in utero therapeutics. This is something that I had never seen before. This might be a brand new session. I don't know if it was at ASGCT last year and I just missed it. In utero therapeutics, all right, making cell and gene therapies for babies. And in particular, there's one with regards to spinal muscular atrophy, actually being able to give that gene therapy in utero before the baby's even born. That would be wild. That'd be super cool. I hope that works. But additionally, this is just a wild one. I'm putting it in here. I don't know what this is going to look like. But there's some guy called Alan Flake from CHOP. And he is doing a session on an artificial womb to support fetal development and manipulations. An artificial womb. I gotta see this. I don't know what's going on there. I need to know. So stay tuned for that one. Yeah, Alan Flake from CHOP. What a lad. Okay, and then another area that I think is really fascinating that doesn't get a lot of attention either, right? Infectious diseases is one. But another component just of our own biology that I don't think gets a whole lot of love and is going to become a much larger section of the field in the future, I believe, is the epigenetic component of our genetic information. Programming our epigenetics to influence the way that that different genes get turned on or get turned off. And so there's going to be a session on that from 3:30 to 5 on Thursday, covering programmable epigenetic and RNA editing for precision regulation. Epigenetics is going to be huge. I don't think this is getting nearly the attention that it deserves. Watch this space, and maybe this is where the people inside of Cercio Bio are just coming in and saying this is going to be a huge spot here on YouTube. Okay, they have a Tuesday poster presentation for Cercio and a Friday oral presentation at 4:15. Okay, thank you, Bart170, for just putting all this in. You all have asked, so I shall answer, and I'm going to get over to I'm going to try to go see their poster number. Bart170 on YouTube, if you can give me the number of the poster as well, that would be greatly appreciated. But yeah, this has been great. Folks, if you are watching on LinkedIn, we are now also over on YouTube at the Biotech Voyager. And we have all sorts of additional content that we're going to be putting on that into the future. Most of our live streams, aside from like one-offs like this one, are going to be living on YouTube. So if you are on LinkedIn right now and you want to see more of our stuff, go over to YouTube, the Biotech Voyager, and sign up there. We've got all sorts of really cool content planned there. We can just put so much more on there. We can go into greater detail on there. So please follow us over on YouTube, subscribe, like, comment, all that fun stuff. So yeah, if you're on LinkedIn, go check us out on YouTube, the Biotech Voyager. It's a lot of fun. But then additionally, we've still got a couple more posts, and we've still got a few more presentations to get through here. Friday. Okay, finally, Friday. Friday is a big one. It's the last day. Everyone's tired because they didn't get enough sleep. You should get more sleep at conferences. Going to bed early is a great hack. Engaging with ARPA-H. First thing in the morning, 8:30 a.m. to 9:45 a.m. Engaging with ARPA-H. Guys, if you don't know what ARPA-H is, ARPA-H, dude, they are going absolutely gangbusters. They are a government agency that are funding pragmatic moonshots inside of biotech. They have funded some really cool stuff. And so it's kind of this government agency inside of the US. I wish we had something similar up in Canada. But inside of the US that looks at what is going to be a moonshot inside of biotech, and let's give some government money, serious government money, to this. So Dan Gibbs is going to be there from Cercium Biosciences. One of the key people inside of ARPA-H is going to be there. Terry Flott from UMass Chan is going to be there. This is going to be a powerful discussion. If you attend one session, I know it's early in the morning, but 8:30 to 9:45 on Friday is just going to be wild. Go come to this one. It's in the grand ballroom, so you can't miss it. Concourse level. Come on, fireside chat. ARPA-H is doing some wild stuff, and if you're trying to build a biotech inside of the US, you definitely should be looking at what they're doing. It's pretty wild. Okay, a couple more here left as we round out the final day of ASGCT. Exosomes, VLPs, and nanoagents. Other modalities inside of gene therapy that don't get nearly as much attention, but they've got some pretty cool stuff that's going on. And so there's a session dedicated to this from 10:15 to 12 o'clock Friday morning. If you slept in and you missed the ARPA-H, first of all, how dare you? But if you happen to get in a little bit later in the morning, this should be the first one you stop by in. 10:15 to 12 p.m. That's in the Commonwealth Ballroom, ABC, in the West End. Interesting. Okay. All right. Another one at the same time is engineering blood cells. Okay. This one's interesting, right? Because we all have blood cells, and if we could engineer them to work better, there's all sorts of different blood disorders. That would be a good thing. So in particular, there's an adenoviral vector in an in vivo selection strategy from Ensoma that looks really, really cool. From 10:15, it's at 11:07 to 11:33 on Friday. That one looks really cool. And then finally, and here's another one that I imagine the folks over at NanoMosaic, Chimin is just off of the camera over here. It's at the end of the day. It's 3 o'clock to 5 o'clock on Friday. But this is new insights into AAV genome biology. Okay? Turns out there's probably a lot of things that we didn't know quite as well as we should about AAV genome biology, and being able to map that all out using something like what the NanoMosaic folks have with the TESSI system probably will help. But anyway, these folks from 3:30 to 5 o'clock are going to be looking at some of these different components of AAV genome biology. Definitely worth checking out. Excellent. Okay, I have been yelling now for over an hour. John Boyce is probably going to start getting mad at me, using the top of his, Oh, he came back and he says he's not mad. Okay, perfect. Well, folks, this is the background that I've been able to bring the ASGCT show to today. Thank you so much to John Boyce over at 28 Capital for providing this amazing venue. Week and it'd be great to say hello to all of you lovely folks. So we're going to end there. Oh, I'm just seeing here at the end Bart 170 is saying 1095 on the website for the Cercio poster. So excellent. We'll look forward to checking that out. Folks, we're going to end it there, but thank you everyone for coming today and for being part of the conversation. Really excited for this week. And just so you know, stay around for just a second, we're planning on doing a post-show recap on Friday. The timing and location of that hasn't been determined quite yet, but stay tuned for that. We might be able to do it on the live floor at ASGCT. Or maybe popular demand says we'll come back here. Maybe John will let me come back. We'll see. But anyway, folks, we'll leave it there for now, and looking forward to just bringing you more content from the conference in the coming days. Peace out, folks. See you later.